Thank you everyone for joining us today in our first talk for the LatinX speaker series. My name is Ana Maria and I'm originally from Colombia, and I'm one of the co-chairs of the affinity group LatinX at the Broad. And the main purpose of the group is to be able to build community and empower the members of our community, especially in the field of STEM, where Latinos make up about 8% of the workforce. It really makes it important for us to highlight and create these spaces where we can showcase the contributions that we do daily to the field. So our hope for this series that we're starting today is to be able to create a platform where we can, as I say, showcase the diversity and the contributions of our community, as well as also create opportunities and spaces for people that are joining to foster collaborations, as well as being an opportunity for networking events. Before moving on to the rest of the agenda too, I also wanted to thank the organizing committee for all the work that they did behind this to make this a reality for today and for the upcoming talks. And also very important thanks for the continued support of the Office of Diversity and Inclusion at the Broad. Without their help, we will not be here today. So in that note, I just want to say that we will now be hearing a few remarks from our Chief Equity Officer at the Broad, René Salazar, and then we'll hear from our speaker. Thank you, everyone.
All right. Well, thank you, Ana Maria. I appreciate the introduction and the opportunity to say a few words this morning, or actually, gosh, this afternoon. The days just fly by very quickly. I think folks on the call know me, but in case you have not had a chance to hear me speak, or we haven't had a chance to interact, I'm René Salazar, the Chief Equity Officer here at the Broad, and I just finished, I think, my 6-month anniversary. So still, relatively new to the job, but really excited to be here. I've had the great fortune of speaking and saying a few remarks at other events for the LatinX group, and I am just super excited that we have Paola here today, who's going to talk about her work. And as Ana Maria said, I think it's really important for us to think about how we as a community can continue to support each other. One of the things that struck me, both in my work in Austin and in San Francisco, is really the lack of representation of Latinos, Latinas in sort of the scientific workforce. And these issues are not unique to the Massachusetts area. I've shared these numbers before, but back in Austin, Texas, where I was at Dell Medical School, when we looked at the Latinx population and the workforce, specifically physician workforce, there was this huge discrepancy. Latinos made up about 35% of the community in Austin, Texas, but only about 5% of the physician workforce, 5%, right? And so when you've got a gap that huge, it's a problem. And so for me, as a first-generation college student, a Mexican-American from the border of South Texas, this is a big problem. And it's something that's so personal to me, and that I think will resonate with all of us in this space. And then when you think about biomedical science through the PhD programs, I mean, the numbers just, they're nowhere near where they should be. And it's not because we are not smart enough. It's not because we don't belong in the spaces, but I think it's about opportunities. And so, what our goal here is really to begin some conversations about the work that we do. And it's really to start to build this community. I will say, even though the numbers here are small, our numbers are small, they're mighty. And I really, really am excited about the work that Ana Maria, Rafael, Ricardo, and others have put into making this a reality. And so I hope that with kicking this off today, that we continue to gather. My hope is that as we think about the pandemic, and it's not over, we know that, but I think we are hopefully going to be able to gather in person soon. I know we already started to demask a little bit. And so, my hope is that we take every opportunity to connect as a community. And it's important that we see each other, that we hear each other, and that we support each other. And so with that, I'm going to hand it over to Ricardo, who was going to introduce our speaker for today. Really excited to hear more about the work that you do, and I'll hand it over to Ricardo. So Ricardo, with that, it's all yours.
Yeah, thank you, Renee. So I just wanted to introduce the speaker and give her as much time as she needs to speak. So Dr. Paola Giusti-Rodriguez grew up in San Juan, Puerto Rico, where she earned her BS in biology at the University of Puerto Rico, Rio Piedras. She completed her PhD in Cell and Developmental Biology at Harvard University, and her doctorate research focused on the molecular basis of neurodegeneration. Her postdoctoral research was with Dr. Patrick Sullivan at the University of North Carolina, Chapel Hill, where she employed the multi-panel collaborative cross-mouse population to better understand antipsychotic side effects. Her research focuses on the intersection between neuroscience and functional genomics, and she was awarded the KO1 Mentored Research Scientist Development Award by the NIMH in 2016. She's also one of the founders or the co-founder of the LatinX, the Latin American Genomics Consortium. And she's currently at the University of Florida at the Center for Adult Research Division, where she conducts research on genetic psychiatric illnesses. So I'll hand it over to Dr. Giusti-Rodriguez.
Thank you so much, Ricardo, and thank you for the invitation. I'm very honored to be the first speaker of this series, and I hope it leads to growing collaborations and networking for Latin American and researchers in the area. And so, as Ricardo mentioned, I'm originally from Puerto Rico, and that is really one of the motivating factors between the work that I'm going to be talking about today. So, this is the title of my talk, and I just want to kind of give a shout out to the members of the consortium and the co-founders and co-leaders. And so there's a big pre-problem genetic study and genomic studies, and which is the lack of diversity and the ancestry of the populations that are being studied. These are two graphs from a paper by Alicia Martin, who's at the Broad and MGH, and it shows that even though European populations constitute a small fraction of the global population, they are overrepresented in genetic studies such as GWAS. This is on the GWAS studies are on the left, and the fraction of the population is here on the right. And we can see that maybe from 2010 to 2018, or 2014, there was an increase in the number of Latin American samples that were on this study. But at some point, we reached sort of like a plateau, and it hasn't really grown since that point. And this becomes a serious issue when it comes to how these types of data sets are then applied in the clinical setting. And this is shown on the right, where if we derive this polygenic risk scores that I'm going to be talking about later, in my talk, it really matters what is the composition of the ancestry of the populations that are studied for the GWAS from which this data is derived, such that if you have a GWAS that is majority coming from European individuals, it's good at the specificity or like the accuracy of detecting of the PRS in an European population works well, but as you're moving to more other admixtures and other ancestral populations, your accuracy really drops down fast. And so, this can lead to exacerbating already existing health disparities in underserved communities and populations, which most of them are of non-European ancestry. And for GWAS, we know that size matters, the type of the size of the samples that you study. So thousands of cases are really needed in order to pass that inflection point, at which point we can start identifying significant findings. And for complex traits like schizophrenia, major depression, bipolar disorder, in the case of psychiatric disorders, it took some time before they were able to accumulate enough samples in order to identify genomic loci that lead to increased risk for these disorders. And so this really, in order to move from this dead zone to the inflection and have exponential growth in the terms of the yield of the GWAS, you really need consortia, because at the individual investigator level, it's really hard to acquire these larger cohorts. And that's where consortia efforts come in, such as the Psychiatric Genomics Consortium, which was created, I think, about almost 15 years ago, and has been really key, together with other similar consortia, at increasing sample sizes for GWAS studies. And one of the first complex, psychiatric complex traits GWAS was for schizophrenia, and this was thanks to the efforts of the PGC, and it has led to increased progress in terms of the success of psychiatric GWAS in the last 15 years. And this has yielded many potential venues of research across many disorders. But the majority of these findings were exclusively focused in European populations, as I'll outline later. And thankfully, more recently, there's been an effort to really diversify these GWAS and expand these studies in other non-European populations. And here I'm just showing sort of the lack of diversity in investigators and samples. This is, I think it's improving in the last few years. This is from about 2 years ago. And on the map on the top, I'm showing sort of the origin for many of the investigators that were part of the PGC. And on the right and the bottom, I'm showing sort of the origin of the samples that have been analyzed. And if we look at the across psychiatric disorders, we can see that there's a really severe underrepresentation in terms of individuals that are represented. So the different ancestries and populations that are represented across samples. So for schizophrenia, there was a recent publication that was included East Asian individuals and populations, but the majority of these disorders the majority of the findings are coming exclusively from European samples. And so, this is like something that needs to be remedied, and that has an impact on the subsequent steps also in terms of data analysis and follow-up work. And really there's a clear need to account for ancestry at every level of the study. Of psychiatric genomic studies. It starts with from recruitment efforts, focused not only in aggregating samples, but also supporting recruitment efforts in Latin American countries, right? So the target population needs to be assessed, and you have to have strategies that are effective for the populations that you're interested in recruiting. The assessments need to be crafted or adapted to different languages and cultures, and have to be tested and with different populations. When recruiting within Latin America, there's a diversity of languages and cultures, and so it's important that it's not a one-size-fits-all at this step. Sometimes it doesn't even work to just translate. It needs to be like culturally adapted as well. And so then when you move to the SNP array, for many of the SNP arrays that are available, may not contain variants that are more prevalent in other populations. So it's important that you include you select the right array that's going to be useful for identifying variants relevant to the populations on their analysis. This also is important when determining what reference panel you're going to use, if it's representative of the populations that you're studying. Again, because it matters sort of like what the tools that you're using to analyze the data. And for a long time, a lot of the samples that had complex admixtures, such as the majority of Latin American individuals who have a mixture of different ancestries, were excluded or thrown out when analysis were being performed, just because they were difficult to analyze due to the complex population structure. And so there's been a need to really develop new statistical methods and modeling in order to be able to accurately analyze these samples and include them in the analysis of large genomic studies. And this will impact, right, like the yield and the findings for the GWAS studies, depending on what populations are included. That's going to impact the results that you get from the GWAS. Then when we move from the GWAS to the functional notation, it also matters, the majority of the samples that are used for many of the functional genomic studies that are then the data is integrated with GWAS tend to come from, again, mostly European individuals. And so how, data coming from expression studies, et cetera, how can we, really harness the information that they can deliver? It's also relevant to consider the populations that are being included. And as I mentioned, polygenic risk scores and their clinical applicability really depend on what populations they're being, the data, what data it's coming from, and what population you're trying to apply the data. And so just to talk a little bit about the potential use of polygenic risk score as a clinical biomarker. PRS is calculated based on a group of independent risk variants associated with a disease or phenotypic trait, right? And so there we grab, right, like all the risk alleles in each variant is summed and weighted by their effect size. And then the result that you get is a single score of an individual's genetic load for a disease or phenotype. And it is actively being used as a clinical tool to predict the risk of developing disease based on your genetic load. But it is dependent on what is the data that is kind of being fed into this number. And so if you're an individual of complex admixture, and the PRS, the data that is being fed to a PRS is majorly coming from European individuals, or a GWAS that is from European individuals, it may not be applicable, or it may yield an inaccurate score. And this is just to show, again, just to emphasize that talking about the ancestry category or distribution of studies that are in the GWAS catalog, as well as the ancestry of the individuals that are present in the GWAS catalog, just to show that it is by far, it is overrepresenting European individuals. And that, from 2005, this is from 2000 to 2017, with you saw this exponential growth of studies, we saw a very small growth in non-European individuals in GWAS. Again, going back to PRS, so it's the clinical application really depends on the genetic similarity between the discovery cohort, the vast majority of whom are of individual ancestry, and the subject under study. And the population differences in PRS are significant. So the risk prediction may be overestimated or underestimated in non-European populations. So it's really important to include multiple diverse populations in the GWAS, and this can lead to an increase in power, will reduce the risk of false positives, and improve the accuracy of PRS in non-European populations. And this is a panel from a paper in 2017 where they looked at how this impact in the accuracy, how it leads to either a misprediction in terms of height, in terms of schizophrenia risk score, and how having a single ancestry versus a multi-ethnic score, how that all impacts the accuracy of the PRS. And so with all of this information, me and together with other colleagues and that are in psychiatric genetics, we were trying to figure out a way to at least work together in Latin America to try to remedy some of these injustices and lack of access, et cetera, for Latin American individuals. And so we got together in October of 2019 at the World Congress of Psychiatric Genetics and established this Latin American Genomics Consortium. We started with about 25 individuals at a happy hour, and since then we've grown to over 100 active members representing 8 Latin American countries, Puerto Rico, and the United States. And we're affiliated with the Psychiatric Genomics Consortium. We have access to both US-based cohorts and Latin American-based cohorts to our members. And the aims of the consortium are manifold. So we want to increase sample size of that represents the admixtures present in Latin America. We want to increase the recruitment and genotyping of samples across Latin American countries. We want to work together to develop better methods to be able to better analyze these samples with complex admixture. And importantly, we're also really invested in facilitating education and training of new investigators in Latin America for psychiatric genetics and genomics. One of the major goals of the LEGC is to bring together and empower Latin American psychiatric genomics researchers. And as you can see on the top map and bar graph, so far we have been able to recruit investigators from 7 Latin American countries, Puerto Rico and the US, which I think is 8. And our goal is to continue to grow this network into additional Latin American countries and the Caribbean. The map and bar in the bottom show the academic affiliation of our members. And this is detailed on the list on the right. And I think we have through word of mouth, we are continuing to grow pretty rapidly. And through efforts that we've been able to facilitate in terms of collaboration, just as kind of like led to more people being brought into the group. And we also aim to have equitable representation in our membership. So we want to make sure that we are including not only, established faculty members, but mid-career, early career investigators and trainees, both at the PhD, postdoc, and other investigators in the master's level students. The graph on the top shows the distribution of academic titles of LAGC members. We have the distribution of the trainees, and we were happy to see that we have equitable representation across genders in our membership. And LAGC members have diverse research interests, across psychiatric genomics, as evidenced in the bottom two graphs. So we have an interest across psychiatric phenotypes and disorders, and both at the identification of genetic variants, functional genomics side of things, more on phenotyping, transcriptomics, etc. And so we want to, attend this when we have to consider all this when we're thinking of moving forward. Sort of what are the interests of our membership. We carried out network analysis of publications where one of our members was either the first or the last author. And many of our members were, are established investigators that have been collaborating for a long time, or they had longstanding collaborations with investigators in the US and have been in multiple publications together. But we want to kind of strengthen these partnerships and grow these partnerships and make sure that they are being kind of moving from like established investigators to younger investigators. And so I mentioned we started, thankfully, we were able to have this first in-person meeting in 2019. We were supposed to have a first in-person symposium in, I think it was April of 2020. Obviously that didn't happen. And since then we were able to secure an administrative supplement from the NIH, which helped us kind of like dedicate time and grow this consortium over the past 2 years. Since then, we participated in virtual symposia from the WCPG. We've given talks in thanks to Zoom, in Colombia and Chile and Mexico. And recently we secured a grant in Puerto Rico where we're doing genetics and epigenetics work with local investigators. And now I think because we've advocated so much about the need for these type of initiatives, we also gain representation of the LAGC members in the PGC coordinating committee, which makes many of the decisions of the consortia. And they have kind of like a seat, we have a seat at the table in terms of how the consortium is going to be moving forward and in driving pushing the need for more diversity, not only in the membership of the consortium, which needs it, but also in the samples that are being assayed by investigators across the consortium. And we have monthly meetings where our members present their work, and we've tried as much as possible to have good representation across. We have speakers that come from the US and Latin American countries across training levels. So we had graduate students, master students, postdocs, faculty present, and representing various niches from genetics, genomics, methods for the analysis of the data, even to talk about specific cohorts. And we even have members and have speakers that don't even work on psychiatric disorders, but they do work on Latin American populations and face some of the same barriers and issues when analyzing these complex admixtures. Okay, and then much of the energy over the past year has gone towards establishing partnerships with potential external collaborators and gaining access to relevant existing data sets. Because many US-based data sets, such as the study of Latinos or the Hispanic Community Health Study, PAGE, the Million Veteran Program, and PsychEmerge on Emerge, they include either biobanks or samples that are of diverse ancestry and will include Latin American individuals or Hispanic individuals. And so we are have been gaining access to these samples. Okay. And in terms of data access, so we're currently working on data aggregation efforts. So we're trying to bring together as many psychiatrically relevant LatinX data as possible. We're happy to say that we have already obtained data access to several large-scale data sets with Latinx or Hispanic individuals, including the Million Veterans Program, the Study of Latinas, and the Boston Puerto Rican Health Study, as well as the Mexican Genomic Database for Addiction Research, and the UK Biobank, which has a small sample set of Hispanic of individuals of Latin American ancestry. And then we are gaining access to additional data sets. We also have access to other independent cohorts that have been contributed by members of our group based in other countries in Latin America. And so we believe that this will give us enough, a large enough sample of about 100,000 individuals of Latin American ancestry that will let us analyze a number of psychiatric phenotypes. We're starting off at the moment looking at alcohol use across many of these data sets, as this is a phenotype that's collected in many instances by these cohorts, and this data is rarely available. And so, as I mentioned, we're working on alcohol use, and that includes a number of categories, as well as smoking, which is also another phenotype that many times is collected across the cohorts. And these are sort of the sample sizes that we are looking at right now, samples for which we already have gained access. And the plan is to carry out meta-analysis using both standard and or local ancestry analysis, using tractor GWAS, analyze multiple phenotypes, carry out genetic correlations, look at heritability, and eventually be able to look at a multi-ethnic polygenic risk score. And we are particularly motivated to work towards having good coverage of our participants across Latin America, both for the equitable representation of researchers, but also to try to capture the immense genetic diversity that is observed within and across Latin American cohorts. And this is showing, that as depending on where you are in Latin America, you can observe diverse ancestry with widely varying degrees of European, Native American, African global ancestry proportions. And this is shown in this figure from this review. And you can see that if we move from the left, like where you have Canada with majority European ancestry, across Central America, South America, and the Antilles, you see this really large difference across the different populations in terms of the proportions of ancestries. And so, we keep this in mind when we're working with different data sets and we're carrying out analysis on these initial phenotypes that we're analyzing. As I mentioned, we already started looking at some cohorts and some of the phenotypes of interest. And we started with this Mexican Genomic Database for Addiction Research, or MXGDAR, and the Boston Puerto Rican Health Study. The cohort at the top is a population-based sample from Mexico recruited in 2016. And the Boston Puerto Rican Health Study is based on older Puerto Rican individuals residing in Boston. And so we have these are kind of preliminary admixture plots, where you have these three ancestries that are included, and you can see that this illustrates sort of like the component of global ancestry proportions for the individuals in each cohort. And you can see that for the Mexican cohort, you have a very large fraction that is of Native American ancestry, versus for the Boston Puerto Rican cohort, you have a larger African ancestry component. And just as here, I wanted to also show an example of why it's important to include diverse populations in GWAS. This is an example where including multiple populations in a study led to the discovery of novel genetic variants. And so this paper by Henry Kranzler and colleagues is an example, of the value of these, of including this multiple cohorts. So they conducted a GWAS of alcohol use disorder using the multi-ancestry million veterans program sample, which includes over 274,000 individuals. And they identified 18 genome-wide significant loci. And importantly, they showed that they were able to identify variants that were shared across populations, but also they identified variants that were uniquely found in specific populations. And so having this diverse sample set led to a more thorough set of findings. And again, due to the complex population structure of admixed populations, samples from admixed individuals have been mainly excluded from genomic analysis. And one of the LAGC co-leaders, Dr. Elizabeth Atkinson, who used to be at the Broad and now is at Baylor College of Medicine, she recently developed a method called Tractor that facilitates the inclusion of admixed individuals in GWAS studies by leveraging local ancestry in these individuals. And so Tractor generates ancestry-specific effect size estimates, can boost GWAS power, and improves the resolution of association traits. And we are actively implementing the Tractor pipeline for the analysis of psychiatric traits in Latin American populations for our studies. And in terms of ongoing work, so we are, with the money from the consortium that we couldn't use for any of the planned travel that we had over the past 2 years, we were able to, we are genotyping over 4,000 samples of LatinX ancestry with psychiatric disorders, including schizophrenia, bipolar disorder, substance use disorders, and controls. As I mentioned, we are in the process of, we have already acquired access or in the process of acquiring access to various cohorts that include Hispanic or Latin American individuals. We are expanding data access to other cohorts that exist that have data available. And we are working on meta-analysis of GWAS of alcohol and smoking traits in Latinx populations. Our work also includes methods development to analyze admixed populations, such as implementing the Tractor pipeline. And we are currently working on a review that would summarize sort of like studies and methods in psychiatric genomics across Latin American countries. And as always, we are working and creating new collaborations with researchers from Latin America and globally. Let me see how I'm doing with time. Okay. And moving forward, we want to continue to address the needs and concerns of our members. A few months ago, we sent out a poll to the LAGC members asking what were their specific needs, in their country that will help move psychiatric genetics research forward. And here I'm showing a word cloud, of the frequency of words used in their responses, which can help us guide us in how to move forward. And, it's the main, the main thing that people mentioned was the need for having more funding to carry out the research, but it's also really important to have fruitful collaborations, to have capacity building, and have the appropriate computer infrastructure to carry out these studies. And so, we are going to make a particular effort, to invest in these areas going forward, and specifically ensure that, all partner members in the LAGC can feel like they're being meaningful contributors to the analysis and publications in which they are involved, and, they're not only involved in the initial cohort collection. And so, if we're thinking about, one of the biggest issues that exists with establishing collaborations between high income and middle or lower income countries has to do with economic disadvantages or disparities in terms of the funding that the government provides or governmental investment in research. And that creates a power differential, between collaborators that are in this higher and lower income countries. And so, and unfortunately, there has been, countless examples of scientific colonialism and bad experiences between Latin American investigators and external collaborators. So, it's really important to follow some, basic pillars regarding equitable partnerships, such as, being able that you identify and invite experts. So that if you're from a high income collaborator, that you identify and invite experts, so you're proactive in seeking collaborators, that from the beginning, it's clear that you are full partners and that you're not trying to be sort of like a data harvester, that you respect data ownership and regulations from the specific countries and the institutions that you're collaborating, that you offer adequate financial support so that investigators, can actually participate in the research that they've been tasked with or that they volunteer to participate in, and that proper credit is given to collaborators in these type of partnership as well. And really to provide equal opportunities for advancement, because there are many limitations that exist, across these different countries in terms of money, infrastructure, etc. And so that is my talk. So I thank you for your attention and for the invitation. And I'm happy to answer questions that you may have. We meet as I mentioned, we have our LAGC virtual meetings on the second Wednesday of the month at 1 p.m. There is no requirement to join our call. So if this is a topic that you're interested in, we welcome you. And if you want to reach out to me, or I'm showing you here, Jenny Samontal-Ortiz and Elizabeth Atkins are the co-leaders of the consortium. We have a Twitter and we have an email that you can reach out if you have any questions as well. That's it.
Okay. All right. Awesome. Thank you, Dr. Paola Giusti-Rodriguez. So I'm going to open the floor for questions. Another item is that we have a survey. So I'm going to put the survey in the chat. And if you can please fill out the survey, we'd be very happy if you did. We had one question in the chat so far. And the question is from Alex. And it says, you mentioned the importance of having genotypic information from different ancestries. Given the fact that the data, the European data has so much statistical power and it's so big, do you know if there's a way to translate and use the European ancestry data as well?
Yes. So one of the things that's become apparent once people have started to implement some of these methods is that you need less sample. So if you have samples that have complex ancestry and samples of different populations, in many cases, if you already have a European sample, adding just a small number of samples from these populations can add a lot of value, and can both boost the European sample as well as the admixed or non-European sample. And so that is one aspect of it. So because you already have sort of like some findings there, you can boost the findings, and you can leverage sort of like having that admixture for your benefit. And that's sort of like what Tractor does in a way. And Tractor, the original paper, I think it was mainly focusing on a 2-way admixture, but it's been adapted, I think, up to like a 5-way admixture.
So I think we should mention that this is an active area of development as well.
Absolutely.
Yeah. Another question I had was, you mentioned using, it seems like the focus is really on the genomics portion of it, but are you collecting different modalities of data? And given it's different modalities of data, there's probably hurdles. Like if you wanted to do MRI, it might not be accessible in different countries.
We are starting with genetic data and genetic samples. I think one of the issues that we face is, for example, in terms of if everyone had electronic medical records, right? Like it would be easier to kind of facilitate these collaborations, but not every country or every system or institution within each country is going to have electronic medical records. And that may hamper something related to like the question you just, am I frozen? Okay. And so we are interested in additional data types. Like we are interested in looking at, epigenetic data and transcription, transcription, gene expression, etc. We're starting with genetic data sets and hoping to move forward once we can secure more funding for these efforts. Yeah. But pretty much if you can do phenotype harmonization, you can look at many different things.
Another question that came up was that how, it's one of the biggest words that came up was funding, the need for funding. It's one thing to do it in one country, but it's going to, you can't control the funding sources in other countries, right? So that's a really difficult barrier to kind of work around. How do you think you can work around that? And then how do you ensure ethical access to all these countries that are contributing?
So the first question, I think you have to include the collaborators from the beginning in the grant writing process so that you can include the resources that they need to do the research in their countries. And so you are correct, right? Like we cannot necessarily count on the local funding for some of these countries because the GDP spending, the percent of GDP that's spent on research is much lower. But if you have an investigator in the US working with an investigator in one of the Latin American countries, from the start, they can be like, how much money do you need to have a research coordinator for fund PhD students, to fund a nurse if you're doing blood collection, etc. Instead of just trying to grab samples without providing funds that can cover all of these costs. For the second question, in terms of access, it's governed by each country, right? Like the data access policies, but the way that we try to do it, which is following what the PGC had done, is that the country and the person that collects the samples, they are the owners of the samples and the data. And so we can establish a material memorandum of understanding where they are sharing the data, but they determine how long they will share the data, how long the samples, or a portion of the sample can be stored outside of the country, and when it needs to be destroyed. And they participate in if it's a paper that's going to be part of the consortium where you have, let's say 50 investigators, and that's the primary publication of that effort, they can continue publishing whatever other studies they want with their samples and without having to ask anyone else, because they are the owners of their data and their samples. Does that answer the two questions? Yeah. So I think there has to be really clear policies from the start. And there's things that we can resolve, right? Like we can't, there's no grants here that will help us build a new genomics core in in Peru, for example. That's not how funding in the US works. But we can provide access to maybe the computing clusters for free, right? So that they can do their computing in the cores that are being paid for by grants in the US. And we can provide training in our labs. So we can have PhD researchers that are PhD students that can start their PhDs in their countries. They can come learn a technique and an approach, and then they come back and bring it to their groups. Or we can host senior scientists as well in our labs. We can help create courses like Cold Spring Harbor-like courses, but that are based in Latin America, so they're more accessible to investigators. And we can also aid with any language barriers that may exist, so that at least with the bigger populations in terms of Spanish and Portuguese, either translate materials or make resources available that can be used for training of grad students, etc. See a question about social implications of sharing. So we mainly work in the framework of the data protections of dbGaP and the genomics repository, so that it's more about researchers that have to apply for access to data, or that is through consortia or through agreements. So I don't know what to say about the 23andMe. They have a very large, I think I remember seeing something like they have over 800,000 Latin American individuals in their data set. I think it's problematic that it's hard to sometimes access this data, but I think that's an individual choice. And so the way that we do our partnerships, it's more investigator kind of based and ruled by their institutions, IRBs and their country's policies for data sharing and access.
Yeah, just so everyone, if they're not joining or they want to see what the question was. So the question was, what are your thoughts about the social implications of sharing public genotypic information, e.g. 23andMe, or collaborating with maybe pharmaceutical companies? They usually have lots of data they don't really share unless you're investing.
We're more definitely on the academic side of things. We have applied for access. We have applied for access sort of like to have 23andMe run our analysis on their samples because it's so large. But for now, we're mainly just focused on academic collaborations.
Speaking about academic collaborations, have you looked into leveraging data from other large cohorts? Are you thinking about biobanking in the psychiatric genome, in the LatinX Psychiatric Genomics Consortium? Because that's, from what I've understood
absolutely.
about Genomics England, that's really, really useful to have samples stored.
So I think but the thing is because there's so many countries, I think it has to be so we would like every country to have their own biobank, and that has for in our case, a psychiatric disorder focused biobank. But that has to be, again, we can't be like, hey, can you guys do this? But I think many of our investigators this is something that they're interested in, but that requires governmental, funding to do it. I know that there's several countries that have a biobank. So there's this other consortium called Latin Genomes, and there's this other group in Argentina that now I'm blocking out on their name, but there's an Argentinian biobank. It's not psychiatric disorder focused, but it is a biobank. But this is something that is necessary. And at the very least, we want to facilitate the investigators that are part of the consortium building a large enough sample size through collaboration so that they can have a large enough cohort to do studies of their interest. And having, brain bank or a biobank, I think will be ideal for sure.
This is one more question. So one of your interests is functional genomics. Is there plans to extend this work into functional models?
Yes. So I think ideally, right, we would do if we're successful in being able to gather enough samples and run our analysis for these various traits in exclusively, Latin American admixture individuals. It'd be great to be able to do this functional genomics assays in samples that are from also Latin American or Hispanic individuals. So to see what is the interplay of the genetics and the genomics. We are trying to do that. For example, the grant this pilot grant we got in Puerto Rico, where we're collecting DNA. So we're collecting saliva, and we're going to be doing epigenetic and genetic analysis. That's but it's a small pilot study. But the idea is to look at the interplay of environment with functional genomics and genetics data and the various phenotypes. For sure, that is ideal. I think it's just, again, you need money to do all this. Yeah,
resource intensive.
But it'd be great. I think all of these areas have. I think there's a lot of need for diversification. So the cellular models that are being used to study psychiatric disorders, the type of cohorts that are included for genetic and genomic studies, all of these needed.
Okay, so I just want to thank Dr. Giusti-Rodriguez again for her talk, and we have another one coming up. So hope to see everyone there. All right.
Thank you so much.